I'm Principal Sheep. Welcome back to our classroom. Before we start, please like and subscribe. Today, we will be learning about the different types of food tests. First, before we learn about food tests, we need to learn about how to make a food sample. You always need a food sample for tests, and this is how you make one. Get a piece of the food and break it up with a pestle and mortar. Transfer it to a beaker and add distilled water. Stir it with a glass rod. Filter the solution with a funnel lined with filter paper to get rid of the solid pieces because it could make your test inaccurate. I'll give you a few questions to test how good your understanding is. Why do you need to use the filter paper? Where do you put the ground up fruit? What do you stir the mixture with? What liquid do you mix the ground food with? The answers are, you need to remove the solid bits and you put it into a test tube. You mix with a glass rod and you add distilled water. The first test I'll be teaching you about is the test to see how much glucose there are. First, you need to prepare a sample. Transfer 5 centimeters cubed into a test tube and prepare a water bath that is 75 degrees Celsius. Add some Benedict's reagent, 5 centimeters cubed, into the test tube. Place the tube in the water bath. Leave it for 5 minutes, pointing it away from you. If it contains glucose, it will either become green or yellow, which indicates that it has a low concentration. If it turns brick red, it has a high concentration. If there's no glucose at all, it will stay blue, the color that Benedict's reagent is, or become a different color that isn't green, yellow, or red. I'll give you a few questions. How much of the chemical do you add? How much food sample do you need? What temperature is the water bath? What chemical do you mix with the test tube mixture? If it is red, is the concentration low or high? The answers are, you need 5 centimeters cubed for your chemical, you need 5 centimeters cubed for your food sample, and the water bath has to be 75 degrees Celsius. The chemical you need to add is Benedict's reagent or solution, and if your solution turns red, it has a high concentration. Now I will teach you the test for starch. First, you need to prepare a food sample as always. Transfer 5 centimeters cubed of it into a test tube. Add 2 to 3 drops of iodine in. Gently shake the tube to mix. If the sample contains starch, it will become a bluish black or navy blue. If there is none or very little of it in, it will be closer to a browny orange or even a different color at all. I'll give you a few questions now. What solution do you add? What color will it be if it has lots of starch? How much sample do you need? How do you mix it? The answers are that the chemical you mix in is iodine. The color it will be if it has a high concentration is bluish black or navy blue. You need 5 centimeters cubed and you need to gently shake it to mix. Now I'll teach you the test for proteins. Prepare a sample of 2 centimeters cubed, not 5. Transfer it into a test tube. Add 2 centimeters cubed of Biuret reagent into the sample. Mix by gently shaking it. If the sample has protein in it, it will change from blue, the color of Biuret's reagent, to pink or purple. If there is no protein at all, it will stay blue or become a different color. I'll give you a few questions again. What solution do you add? What color will it be if it has protein inside? Well, how much of the solution do you add? How much sample do you need? First, the solution you need to mix is Biuret's solution. The color it will be is pink or purple, and you need 2 centimeters cubed for the food sample and 2 centimeters cubed for the solution. Next, I'll teach you one of the 2 tests for lipids. First, prepare a sample of the food, but this time do not filter it. Add 5 centimeters cubed into a test tube. Add 3 drops of Sudan III stain solution. Gently shake it to mix it. And since the stain stains the lipids, if the sample has lipids, the mixture will separate into 2 layers. The top layer will be bright red. If there are no lipids at all, there will be only 1 layer. I'll give you a few questions. How much stain do you add? How much layers will be there? What color is the top layer? How many drops do you need to add of it? How do you create the sample? How much sample do you add? The answers are, the stain is called Sudan III stain. There's 2 layers. The color of the top layer will be bright red, and you need to add 3 drops. And you make the sample as usual, but without filtering it. And you need 5 centimeters cubed. The next test for lipids is more popular than the Sudan III stain. They're both accurate tests for lipids, but this one is just simply more popular, and you should probably use it more in your tests. First, prepare 5 centimeters cubed sample of the food. Then add 5 centimeters cubed of ethanol into the sample, aka into the test tube where it should be in. Shake it vigorously, not gently this time. Add 5 centimeters cubed of distilled water. And if there is lipids in it, it will create a white emulsion. If there isn't an emulsion at all, there are no lipids. Now I'll give you some questions. How do you mix it? What color is the emulsion? What chemical do you mix into the sample? The answers are, you mix it by shaking it vigorously, the emulsion is white, and the chemical you add is ethanol. And that is it for today's video. Please like and subscribe if you learned something new. 3, 2, 1.